My name is Abhishek Rathore. I work as a biometrician with ICRISAT, and it's a pleasure to see all of you here. And I'll talk. Well, so from the morning you are listening that how data management we are dealing in ICRISAT. What are the things? So I'll also. Am I audible? Okay, thanks. Yeah, sometimes two mic creates lots of stereophonic voice. So I'll also try to discuss some of the things what we are doing, and biometrics unit here do the work to gather data management unit, all of our readers and scientists across the theme. So usually when I do this kind of presentation, I appreciate if you ask in between. You can, anytime you can stop me, you can ask whatever if you have got any query. Don't wait for the final like when I'll finish. So I try to make it more interactive. So please feel free to stop me any point of time when you like. And it's also my pleasure today. I could see many of my seniors who are sitting here, my colleagues who worked with me in earlier institution. So, and well, many of my ICRISAT friends also, but some friends who earlier worked with me. So it's really a great pleasure to see everybody here. So I have been asked to talk on research data curation, validation and analysis in 1 hour. So normally I take 1 week workshop on this. So today I'll try to finish that in 1 hour. So in general I'll talk more on general aspect, and later on we can discuss if you need any assistance or you want to do any collaboration on the things. So when we talk for data management and data, what is the usual workflow for any data, any project? So I try to keep it general. Like we may have research station data, what we collect through field books, through electronic capture mechanism. Some of my friends can conduct farmers participatory variety trials. Many of us are involved in that. Frontline demonstration, lab experiments, baseline survey. With all of the project now baseline survey, then adoption surveys, some other surveys, field books. My friend Chuka has talked for agrobase and many other softwares by which we get lots and lots of data to our projects. What we do after that? We curate the data. Sometime we do, sometime we do not do. I'll talk more on the curation. So it's then we do the analysis, then we have some interpretation, we publish it also. After that, what is after that? After there are two very nice cases. First, we archive data, store it for future generation. Younger generation of scientists who are coming, they will get benefited from our experimentations. And second case, forget it. Most of the time data lies in the Excel sheets and die with time. So what is the key? Key is the data sharing. Once you share the data, it is with you and with the community always. We need to be open for the data sharing. Okay. Because when we were in my previous institution, I remember I gave most of the time this example of data sharing. You know, biometricians have several roles. Sometimes we need to behave like economists. Sometimes we need to behave like breeders. Sometimes we need to behave like soil scientists. So in my previous institution, I have done a survey, a soil science based survey where we have collected lots of data, like GPS based data of the soil nutrient in 2 districts actually. And we have around few thousand samples we collected, we analyzed. It was a huge project. What happened at the end of the day? After 8 years now, I don't have that data. It's my own personal experience. We don't have the data. Why? Because we never shared the data. But yes, one thing I have from that experiment. Some nice photographs. How? Because I shared those photographs through Facebook. They are still on my Facebook account. So whatever information I shared with everybody, that is only lying with me. All the information has gone. So that is the power of sharing. If you try to relate it, whatever you share with everybody, that data exists. Okay. So mainly two kind of things comes, data capture and storage. We all are expert in this. Well, Chuka has just shown some tools to even enhance the data capture and storage, but we are very poor, not all of us, okay, few are expert, few are doing in data archival and sharing. So this workshop is all about that, how do you archive it, how you share it. Data capture has several formats, like the breeding data management. If we have some breeders around, so those breeders will know. We develop nurseries right now in the data management system now. The days are gone when you do that in Excel or somewhere else. Now we do that in data management system. We develop nurseries. We make the process itself in the software. We develop experimental designs. We prepare layouts. Then we collect from the field data by handheld. Some of them Chuka has shown. I'll also show a kind of handheld. Then the barcode readers are there. You can directly measure the plant number, anything, and then electronic phenotype measurement devices like the green color, the leaf color, leaf texture, and even the plant height. Many things you can just note down by the electronic gadgets attached to your handheld tools. So this is then again if we come to socioeconomic survey, because I can see many of the social scientists are sitting around here. They also have some templates, and these templates, then they have the survey data tool. They collect the data from the field, like they go, and then sometimes they also use handheld devices, many of them, and to collect some market information. Sometimes they use barcode readers also, like if they want to read the level of pesticides, they want to read the level of maybe the fertilizer. So authentic information, there is no error in noting. And we are working on the tablet tools where you can record the voice, you can even take the images, and you can store everything, and you can transfer to the databases. So these things are happening. But what happens then? You store it sometime into the database, sometime you don't store it. The key here, you store it and share it, but then suddenly one question comes, what about the data quality? When you are sharing a data from an international institution, it should be free from errors. Errors are of two types, not statistical type 1 and type 2, but still there are two types of error. One error, what is data recording error? I have been told 4, I have recorded 14. That's a error from our side. Second error is the respondent error. My socioeconomic friends know. Sometimes farmer do not want to tell his income. They don't reveal. Many ladies do not reveal their age. Many do not come for the photograph. Like they are the very practical problems. None of them is, they are practical problems when we want to segregate data based on age, when we want to segregate data based on the land holdings. Sometimes people don't declare correct land holdings. They just tell whatever they have visible, sometimes they just hide. So these are the very practical problems. That kind of error we cannot avoid, but we need to identify. If we take the data without cleaning those kind of secondary errors, we are gone. We might find a Bill Gates in each village. They might say their income so high, sometimes they may say their income so low. So we need to, so before data sharing, data cleaning is very, very important. It's very important aspect. I should say sometime I'll go to get some water. But you know, data cleaning is very tricky. It's very tricky to identify what actually, is it a type 1 error or is it a type 2 error? And there is a type 3 error also. You know that, what is that? Respondent has filled data himself. Sorry, yes, the person who has gone to take interview, he filled data himself, sitting in the hotel room. It's a very practical problem. Many of you may not agree, but that happens. Not many times. So to tackle those things, we are developing online tools, as Chukka has shown and has been shown by my colleague earlier, Sumanta, where data will be recorded based on the latitude, longitude. It's not for monitoring, it's for making efforts. We are working for a purpose. Here at ICRISAT we are working for a purpose. And now also we are working to elevate the livelihood of the persons for whom we are working. And we want to ensure that whatever data we are working with is fine. Anyways, so now I'll talk on the importance of data cleaning, but please forgive me, my socioeconomic friends, because the example what I'll take is a breeding example. And I know we all are working for some variety, good variety release end of the day and market linkage. So two people are very important for me, my breeding friends and my socioeconomist. Okay, so I'll take an example from the breeding side, and same I can take an example even from the socioeconomic side, but in socioeconomic the dealing of data is not as straightforward. Do you agree, sir? So how we do the data cleaning, I'll take a breeding example and then you will also came to know its importance. Okay? Any question till here about data sharing or data cleaning? Anybody has any? Because data cleaning is very tricky, and finding influential observations and outliers are two different things. You may have an influential farmer in a village, but sometimes you may find even an outlier farmer in the village. And we know that when we do analysis and we talk for statistical analysis, when we talk for 95% confidence interval or 99% confidence interval, sometimes we may need to even take out the actual observation. Not take out, divide actually. We may need to group them in different classes and then we do analysis. Otherwise, we may end up with doing some analysis which may not be very relevant. Okay, so you are not going to do any analysis during my presentation, no hands-on, only we'll share some thought process. I'll talk with the data in which we have a recombinant inbred line. We had 150 progenies, like 150 plants, 150 genotypes. How many of you understand all agricultural issues, or actually how many of you do not understand these kind of data set? Anybody? Like we have a problem of testing few varieties in the field, and we also had another our idea once to identify which of them were good, and then we genotype that information and look for the genes which are good, which we call as QTL mapping. Few of you might be aware, few of you might not. Okay, so we had this data. It's a real data. From where we took, source is unknown, but it's a real data. I can provide you if you want to have your hands on. And we were supposed to do analysis and share this data. So question came like, whenever data comes to a statistician, we always first do data cleaning. We never directly put the software data into the software and start analysis. No. If we are going to do that, we are really doing some horrible analysis. So if I tell you most of my units time goes only in doing data cleaning, less time goes in analysis. And so I did analysis for this. So now each of you, like, you should share your thoughts, please. Just don't only listen to me. So it was a RCVD design. We had a block factor, we had a genotype factor, we had an error factor, and then we had a total factor. If you could see, we have got highly significant results for the genotype. This point I'll stop for a while. How many do not understand this table? Please feel free. I can explain. Everybody understand this table?
6 decimal point. Why was it?
What?
So many decimal points.
Well, that is another issue. I could have removed them. Thank you. I can do that. But like, do you understand this kind of analysis of variance table? Okay, so this table tells us the sources of variation, like what we are going to analyze. Like the field variation we want to see. We want to see the plant to plant variation, and then the error, and then the total. In the kind of designed survey, when we design some survey collection and we want to estimate the average yield or some average, at that time these kind of statistics are generated. Then you compare whether the total yield of this year of a particular grain commodity was higher than the yield of the last year's grain commodity. Then also you can generate these kind of significance values. So when this value is less than 0.5, that clearly shows that you have got a very good result, like things are significantly different. Okay? But, and this is a very important factor for the breeders and many other scientists. If you see that R square is 0.9, is it good? That you can say in the regression, isn't it? It's a good R square, isn't it? Jupiter. It's a very good R square, isn't it? CV, how many of you understand coefficient of variation? Anybody who understands it? Breeder friends know this. You know? So there is a say that CV should be somewhere around 10 or 15, then trial is good. Huh? We say it, isn't it? And but you know, most of the time why we see CV? We say that it is the repeatability of the experiment. Well, I don't agree. I'll talk later on that. So, but so you see the CV is 4.8, error is 3. My God, very less, and the mean is 63 of the trial. When any breeder gets this kind of analysis, immediately he starts to write, data was significant. When then we do significant comparisons, start to write stuff. But error is high. I think it's quite low. Yeah, it's only 4 digits, and this has the 5 digits. Okay, and anyway, like that's not the aim right now here. Why? Because that error changes data set to data set. If I'll see your data set, then I can explain why it's so high, why it's so low, with you, sitting with you. So the aim here, this looks a very nice analysis. Perfect data. Very low CV, actually 4 or 5% CV, normally we do not get in the field trials. But you know, a statistician is always suspicious about the data, no matter how good data is it. Jupiter. So I always, well, I'll come to that. So, but the data belongs to a very good student. He even did the QTL mapping. Many of those who do not know QTL mapping is identifying the gene in our DNA which are responsible for a trait. Like if I have that gene, I may be tall like Jupiter. I wish. And if I had another gene, I may be slim. Like so those kind of genes, a diabetic gene. So we do the study, and then we try to, from those, we select the plants which are performing high, and then what special is in their DNA in comparison to what is special in the plants which are performing very low. And then we see that significant difference. It's very simple. It's simple regression, but looks high-fi. The identification of a gene responsible and regulating the functional mechanism of something. So, but it is not more than a simple linear regression, let me tell you. Okay. This is also true for economists, okay? So, but this gives a very nice picture. So, what we do, we call for a threshold LOD, Lord score. Normally, this score we generate through some permutation test. So, any peak higher than 2.5 will be a putative QTL, suspected QTL, like there is some effect. So, the student came to me, Abhishek, I got 4 nice QTL, you know, I'm going to get a nice paper in crop science. I said, okay, all the best, but let's stop for some time. Why don't you show me the analysis that you have done and send to me the data? He agreed because in ICRISAT it's a very nice culture. Like most of our scientists are well versed in statistics, but whenever it comes to publication, sending to our, then everybody wants to be very sure and wants to generate the quality analysis, and then they want to check their theory with us, and then we do that kind of analysis. So you know, a statistician never sees the table. We see the figures. We like the figures. So I did the analysis and I say, okay, let me see the distribution. So he showed me the distribution of the data. He said, see, it's perfectly normal. Okay? Sounds good. Perfectly normal. I said, no. We never believe in the distribution of the data. We believe in the distribution of residuals. When I calculated distribution of residuals, many times we do normality test of our data. Data has to be normal, you know. But whether data is normal or not, error has to be normal. That is, the residuals has to be normal. Like, what are the errors? The measurement error, or the kind of differences present between us which we are not able to account through statistics. This was not normal. This has to be a normal. Then I said, there is some problem. Okay. I said, okay dear, you come after 1 week because now I need to investigate the data. If this would have been normal, I could have said, okay. Then we did something else which we call influential observation analysis. It's a bit higher level of statistics where we deal with the leverage and a very popular thing which we call Cook's D statistic. This doesn't look for a Cook data, okay? It was the name of a person called Cook. But fortunately, it can also detect the Cook data. That is the beauty of this. Well, we are not magician, okay? We don't do a stick. We also play with the tools and mathematics tell. So here we have around 500 observations. These 500 observations for one trait. If you look through the Excel table, you can never figure out if you have 50 traits. So these are the plots which they were giving me 2 long bars. They said that there is some problem looks at 2 places. But sometimes these things are correlated. So we need to look for the highest value of the Cook's statistic. Okay? We did it and I thought that somewhere near about 450 or 435 there looks some problem. Okay, I went back to the data. I opened my sheet. And then I was looking into it. Again, there are so many things, it's difficult. But then I spotted that around 443, there was an observation which value has a 17.32. Okay. When I sorted data on the replication, like the same plant tried at other blocks, I could see the all value was 71, 70. This particular value was the 17. So no need to tell when it was being typed. 7 was pressed before, 1 was pressed something. So I asked the student, please come with your field book. In field book, it was clear that while he was recording data, he recorded 71.32, but when he was typing, it's a very obvious error. But if you see my first ANOVA table, 4%, 5% CV, 90% R square, anybody could have imagined this was the problem with that? But you know, we always suspect. So then I changed it, okay, and again I did analysis. You know why now? Why statisticians take this much time to analyze even your one trait? Because we never know that your data do not have any problem. If you will not see it with a suspicion, so all data pass through all of these checks before being going to analysis or going to be shared online. Now, oh my God, R square become 96%. Huh? Breeders are even happier now. The kids got even happier. Hey, Abhishek, you have really enhanced the value. Repeat. Oh my God, CV is 2.6, just half. Remarkable, isn't it? Error was around 3 last time. Error is now 1.6. Yield mean is same. You remember it was around 64 or 65? Still it is same. No change in the mean. So your eyes sometimes cannot see that, but his eyes were popped up when I showed him the QTL analysis. If you see all of the QTL what he got, all of the genes what he got, gone. You see, we got only one, and this one was somewhat which was not detected earlier. Should I show you? You see that this came somewhere here, and you don't have any QTL detected here. It's this much. So we identified some wrong portion of DNA as related to the trait, and that too for many. So the now twofold problem. First, all of these four were spurious. GIGO, you know GIGO? Garbage in, garbage out. Statistical software are GIGO. You put garbage, you get garbage. So, hey, not the statistician, huh? Few of them maybe, but so okay. So now we got this QTL detected, but you know still we just cannot believe the data again. So again through my substation, I plotted the. Now you can see that distribution of data becomes really good, but we never believe it. We believe a residual distribution. Oh my God, it's same. What to do now? Again I did the analysis. Now I see that the younger brother became the big brother now. Okay. Now what is the problem? I went again to the data. Okay. The one trait analysis is going on. Okay. Only one trait. Again look for the data. Now I got another recombinant inbred line number 107. Then two replicate values were 82, 80 and another was 47. Oh my God, this is very difficult. We have to sit with the supervisor of the student. Because last time it was very clear. This time it is not clear. Because two replications are suggesting values somewhere around 80 and one replication is saying this is 40, 45. 50% drop is not real. It is not a real. It can be real situation if all of the genotype in that particular block were half due to the fertility, due to the blocking. Then we could have believed. This is not a believable situation. We actually, the student said no, this is the case. He observed 47 only. Fieldbook also says 47 only. So there was no recording error. These kind of errors are very difficult to tell, whether they are the true value or they are outlier. You get this kind of, in economics you get this full of this kind of data, which is true but can't believe that data. So after lots of discussions, I said that is the reason the statistician forced you to take replicates. Let's treat it as missing. We are not removing the full 107, but there is no harm in taking as replication as missing, because this is a clear case of mislabeling of seeds. When it happened, how it happened, nobody. And because this data was not, that may be the one reason. Second reason, plot might be affected by some disease, less number of plants. Somehow we don't have plant head at that time. So safest way to use this as missing. It's very tricky again. It's very tricky how to deal with such situation. But we can't do anything in this situation. Okay. So I put this value as missing, and now I give this remain as is achieved. I repeat it again, all analysis. Okay. Do you have some thoughts on this data cleaning? So one trait analysis may take weeks also sometime when situations are like this. For the common errors like 17 and 71, we don't go back to the scientist. We know. But for such errors, we cannot take a decision. A statistician cannot take a decision at his own for such cases. Okay. So now if you see, R square is 99%. Okay. 100% repeatability of the experiment. Coefficient of variation, 0.6 from 5 point something. 10 time reduction in the coefficient of variation. Error, just forget it. Huh? Yield mean, no change. So sometimes the mean and other statistics misled you. You don't know what to do, what not to. And now if you see the QTL, this gentleman become even higher, and we got another gentleman here. So now there are two true QTLs, one with lots of confidence more than LOD score 4, and one another with somewhere LOD score 2.5. These LOD scores are like percent significant. 3 LOD means 99.9% confidence interval, and 95 means 2 around, something like that is the correlation. So, how many observations were there which have started the game? 2. So, 2 observations, if not cleaned properly, can destroy your scientific career. This is not a joke, because if we would not have analyzed that data with care, the student would have got 4 QTL. And you know, in the breeding, based on those QTL, he would have designed his next plan to validate that, to transfer that QTL, and he would not have validated or transferred that QTL. His more than 1 or 2 year might have gone, and his PhD would have been extended by 2 or 3 more years. So these are the very sophisticated cases why we need to look data with suspicion. Is it okay? Is it clear? And they are very, very fine, typographical, just very 1 or 2 observations can distort the game. And if we share such data for the open access, you are gone. So that's my responsibility comes, how to clean the data. So this is the thing, how we do the cleaning of data, and it is for one trait. You see, the most of the experiment has 20 to 30 traits, 5 to 10 locations. Then you can imagine the kind of time and resources are being used for data management and sharing. It's not an easy job. It's not a simple job. But since we get money from the donor, the public money, for the public we are working, data need to be shared. We need to invest this kind of resources. And data cleaning has to be very important. So now, again I did analysis. Plots, because I actually see plots first, then the ANOVA. Now this distribution is very nice. Okay, good normal curve. Okay, this I don't care. This is good. And you see our influential statistics. Okay, fine. Now all are raising their voices. Some are raising more, some are higher, but they are not dominating the game as they were being dominated earlier. So these are the very, very important steps in any basic analysis. You can see that residuals are really spread now. There is another statistic we call Cove ratio, which we look for the. So this Cove ratio is also very nice. No problem at all with our data. All situation under control. Okay. Let's try to understand this once again, and let's try to see how things move. Yield P2 is the variable with the 2 problematic observation. Yield P with the 1 problematic observation, and yield G is the last one, the good observation. Okay. PV reduced 8 to 9 fold, R square 9 to 99, error estimate from 9 to, all of your repeatability of your trial increased, enhanced by 10 times. It's not a joke. Repeating your trial just by proper data curation and analysis. Okay. Now, if you see this lower table, when we were doing the using yield P2, we got the 7,101 significant pairs, like all with all. When we do which variety is the best, we do all pairwise comparisons. So, out of total NC2 combination, so we got 63% significant pairs. Minimum standard is 2.5, maximum standard of the difference. That's why it's different from them. Is 2 point from it is FED. By removing one observation, this became 79% of our observation were like you germ plan. The lines you were having were really fantastic, but were buried through one observation. Okay, this is 80%. So 20% significant pairs were buried under one observation. And when I removed 2, 95% pairs were significant. Like you have the student has a fantastic material, very nice, classic, very nice material. See, 95% were pairs were significant different, and error reduced, SED, standard of difference, like when you compare two things, reduce more than 10 times. There is no magic, only good data cleaning and good analysis. Okay, so what impact it has? It has the impact if you are comparing a local check with the improved variety. What would have been non-significant? A significant comparison would have been treated as non-significant. A significant upliftment in the socio-economic status would have been declared as not significant. Because when we talk for economics data Jupyter, then you have so many variables and so much variability. And then I know that the way you deal it with, like, that's why it takes time to you to look each observation individually. Well, sometimes mathematical tools can help you to identify the kind of variation you have, but it's so complicated. So I understand why you guys take so much time. But well, these tools can help socioeconomic studies also to figure out the clusters of the problems. They may not be the problems. Is it clear to everybody now? And this is the profile of the QTL, you see. All these 4 QTL just got disappeared. We got a new one, and this new one become even higher, and we got this gentleman came up with only 2 observations. So never underestimate 1 or 2 observations, because sometimes we say, hey, it's only 1 observation. Let's do another. Nothing will happen. It will happen. Is it fine? No voice, no thought, I don't know. I can understand my colleagues who have come from far, from Africa, from USA, they might sleep. Okay, now I'm not done, okay? So now I just want to talk something on CV. We are very much fascinated for CV, isn't it? My breeder friends, my statistician friends, many are very much fascinated for. If CV is 50% or 60%, hey, discard your trial. But I will say, if you have a 15,000% CV also, don't discard it. CV is a okay type measurement of repeatability, but this is not the absolute measurement of the repeatability. You should never take it as absolute. If your CV is high, don't think your trial is bad. Okay. Then CV is just an indication that there may be some problem with your data. Then you need to look to your data. You need to go for data cleaning. And sometimes even a nice, clean, perfect data can have 10,000% CV. Why? Why? I know many of you are not agreeing with me. You will blast me later. Because CV has mean somewhere at bottom. And based on the trait, mean can be high, mean can be low. And if you have some trait, because X is a real valued function, okay? If it takes value from minus infinite to plus infinite and mean is somewhere near 0, you may have millions value of your CV. It may be negative also, you know. Have you seen negative CV sometime? Anybody has seen? CV comes negative also. Nothing wrong in that. If a journal person, a referee asks you, direct mail to me, I'll answer. Why it's negative or why it's positive. So those are few myths which we need to come out, and it's a good measure to see. If it is high, just look at your data, but just do not believe everything what you see. Okay, is it clear or not? Any question? No question? If not any more question, I will not end. I'll continue. Now I'll also show you briefly a tool which we use for data collection, as has been shown by Chipka, by Sumanta. This tool we have selected for breeders mainly, but it's equally good for socioeconomic science. I'll show you how. This is a handheld tool. So like how many of you do the field experiments? Oh, many. Okay. Yeah, sorry. Like to me, the all person who works, yeah. Yes, even the entomologist, yeah. So such kind of things are a bit tricky. But like to avoid this, there are two issues. Using good handhelds, and now you are also part of now TL3. So in TL3, we will push these kind of data validation tools also, where we can have these things inbuilt. At least some level of things will be in. Yes, electronic. Yes, that electronic data capture is on the and the transfer to the server and to the laptop is on the list that has already been made as some kind of activity, so that you shouldn't have any problems. Same I wanted to show, but somehow it has stopped. It's not obeying me. It's working in here, but I don't want to show you through the projector. Okay, so we have a application what we call field lab. And it looks like a usual tab application. So this works based on a template for our data. Like if we have got a RCBD design, it has template for that. It can take any table with it. Like if it has a row column structure where you have the respondent number in the rows and the traits in the column, we can generate it for that. So like I'll show you a workbook. So it's very simple and straightforward. You specify what kind of workbook you have. You may have a ICIS workbook, you may have an Excel sheet, only a row column sheet. And most of the time we convert that to, so like I have a template in here, and if I want to view it, I can show you. This looks like this, something like this. So this template, I hope Jim you are also familiar with this template. This is the same template what we are using for Aware. So we have tried to make a template which works with Aware, work with our data management system and with the tablet tools. It's very simple. Like it comes with the study title, primary key, some metadata, and the most important thing what you need to tell are the factors. Plot, pedigree, block, genotype, anything can be factor here, what you think is a factor for experimental design. For socioeconomic survey, these things need to be like the respondent ID, the name of the person, the village, which are the kind of fixed information for a particular place. And then we have some other fixed values like pH of the soil or the latitude, longitude of the location, name of the village, which will not fix for that particular survey. So those kind of things will come in here. And then there is another tab called observations here. Here if you see that we have those treatment number, block number, plot number, variety number. Same thing can be replicated to socioeconomic survey. You can have respondent number, respondent name, and anything like, because we in socioeconomic also survey also we recorded two-way table. We have the name and then we have the field. Your name, number of cattle, land holding, male, female, everything, and beautifully all things can be here. You need not to write most of the stuff. You need to select most of this. I'll just show you. So this is where we need to make template. In ICRISAT now what we are doing for our reader friends, these templates we are developing. Like suppose somebody comes to me that Abhishek, develop a survey tool for me. These are my questions what I'm going to ask. Then what we do, we take those questions, we develop these templates for them from the metadata what they provide us. They provide, okay, this is a sample survey in this particular area. We make this template, we import in this tool. So right now we are high tech. When you ask us to generate a design, we say send your tablet to us or send your design to us. They say they don't have design, we generate a sample design for them. Then we convert that into a tablet friendly file. That tablet friendly file you download into this tablet through your email. This works, you know, this has an email, it has a USB connectivity, and then you simply go to a study, click on the description, and you see that same file is right in here. It has a tab called factors, variates, constant. You can just click in there. You can see whatever the factors, plot, pedigree, block. Then you will have respondent number, respondent name, and all other information for them. Then you have the variates, what you want to select, age, height, language, and any other stuff like cropping methodology, crop on which you are working, all can come in here. And then the constant, like the temperature, geographical location of the place, latitude, longitude, all information is right in here. Well, you can change it in here, but like this is the fixed. Now we will go to the observation, which is the second tab. Just click in that tab. And you will get the layout. What this layout is saying? This layout is telling you, oh, I'm advanced. Huh? There is a column for the yield. There is a column for the LSS. This is some leaf, late leaf, some score for the, from the groundnut. Then the rust, and there are some other columns. This is highly customizable. I have selected only 4 fields here, just to show you that what we. So if you want to see more, you can turn on your tablet like this, and then you can see even more fields. All the fields can come. So, and the tools what we have selected like this tablet, you can see it's very bright. You can read in it in sun at 12 p.m. You can record the data, and they are very handy. You can hold them in hand. Something happened. Fortunately, not big deal. Huh? Now, if I'm in the field, I'm recording plant-based data, and like I wanted to record some plot yield, I'll come here. As soon as I'll come in plot yield, automatically a keyboard will come. Yeah, that's the job, to press things hard. Okay, so this is our field layout, and then when you click in here, you can see a keyboard. Can you see that it's a numeric keyboard? There is no alphabets here, like you are recording something which is numeric. It's very intelligent. It won't show you. So you can type in, like I typed 22. Okay. Now when I come to LLS, I click in there, I got a score sheet automatically. It's asking no LLS, very low, low, medium, whatever you observe in there, just click it. I observed medium. I click medium. Then it went to next field automatically. Now it's asking what is the rust scoring? 0 to 10, 10 to 20. I see, yeah, it is not rusted, 20 to 30%. Now you see that all of the information is right in here. 22 you need to type, maybe this LSS and rust you need not to type. One thing it has taken as character, another thing it has taken as the numeric. Okay. Now there was something, the fourth field which was days to flowering, DF. I entered a value 2, 2, 2. By mistake, when I was pressing, I pressed 1, 2 more. It has complained immediately. Value entered not within the range. Okay. Maximum value 22, minimum value 75. How do it get it? I did it. Like when we made those templates, at that time we did it. No magic. Okay. Based on the crop to crop, it worked for you. Like we have got the latitude, some income idea. It will not take any income of 1 million. It will only take the value what is required. Or sometimes for something like the field like male and female, we can make that field. You click, option will come M or F. You just cannot enter anything else. So all data validation is also being taken care right now in here. Number of kids you have, maybe 1 to 10 or 1 to 20. Something like things are fully automated in here. No chance of typing any character because character keyboard will not pop up at all. But if it is asking, do you think 222 is correct? If you say yes, it is okay. I'm boss, I can do, click on OK. It will highlight as is right. That it is saying I'm accepting it, but there is some problem. Okay. It's not as rigid as you thought that it will not take. No, it will take. Okay. So these kind of data validation are inbuilt. But what I prefer that when we did our soil fan survey, we bought a Sony voice recorder like this. I don't know if you know. So that voice recorder was very nice. When we talk to the respondent, we record them all. We ask the consent that we are recording what you are saying. Later on, if we find something really interesting, so we know who told us this story, that facility is inbuilt in this place. There is a recording button. If you will click it in, it will record for each cell. For each cell, you can have a recording. So if you see something interesting in field, you can do the voice recording for each cell. If you are in field, you see that the problem in the field, so you can record what is the problem with that particular place. Or you want to listen to story of somebody, you can record it and write in there. What about photograph? They are also in there. Click on the camera, it will bring out the camera out, and then you can have a photograph of the field. You click it in, photo has been taken. It will ask if you want to save it, just save it, and that photo has been taken. This photo will be saved. You can see that the icon has come here. Like this particular field has a photograph. You can take multiple photographs for each of them. So now, in the data, you have everything. You have the observation, validated observations. You have the images. You have the voice memo. If like for any of the problem, what you think, and you have whole history with you. Voice I can also record, but it works. So don't worry. And once you are done, you just go back. All of data is with you. You put in on a USB cable. It is synchronized with the IVP. I don't know how many of you know the IVP tool. Like with that, it will automatically come to the database. But this is not only limited to field trials. You can use it anywhere. Farmers participatory variety trial. You can use it for the sample survey. You can use it for the lab. You can use it for anywhere, because the templates what we have developed, though they are based on the rectangle work frame, and it works. Okay. So like when you have the table in your sample survey form, table 1, table 2, table 3, all of those tables can come with different templates, and you can, like from the study, you click on the open study, it shows the study. So you can have all studies, like you want to record that particular status, all of them will come. Everything is there. This we are working for CRP 1.1. We have already made templates for them, but handhelds we have not given them as of now. But otherwise, data validation tools are right in there from them. Then you can export data. You can import data if you have recorded from somewhere else. There are something in maintenance that I will not discuss. Like there you can change the what. Well, I can show you if you go to the observations. At that time, you can choose what factors or what variates you want. If you know the Android system, so it has a menu. If you click on the menu button is right in here. Then you can have the factors and variate. You can click on the factors and variate. Like what variable you are recording as of now. Those lists will come. You can check or uncheck. You can specify range right in here. The range. It's very easy, very user friendly. But we never bother our breeder friends. We don't say that you do it. When we generate a design, we have the trade dictionaries, we have the ontologies with us. We give the standard name, standard range to them, and we give this ready to use template to them. We don't want them to waste their time on these things. Like it's not very. So the kind of design we give this template also to them. So okay, friends. I think with this, I'll take some more shots, but I think it's better if Jim shows them. Jim? But I'm not done. Yeah, so once the data has been captured in the templates, then that data can go in. This is the open source tool. Yes. Free to everybody. Anybody, you can just have this tool. No issue for that. Like it can be downloaded from free from the IBP workflow website, or if you can write us, I can also send you the link, and then you can use this. So a few more things like this data will automatically go to aware, the kind of like Jim will show you. Those things are already like in, because the template format is same. Few templates will be given to you tomorrow by Jim. Okay, Jim, I hope we have a plan for that. So we will distribute those to you tomorrow, and then you can use them. And then once data based on latitude, longitude is right in there, you can visualize your data through the aware platform. How to use it will be shown, but you will be seeing such kind of pictures tomorrow, today when Jim will discuss that whatever data you recorded from the field will directly display it here, because it will automatically come to your PC. From PC you directly upload the template, specify the field, the validation, and you can see that from where you collected, what are the different ranges. And then the breeding perspective, maybe I'll show this you later on when Jim will introduce to the system, that how we can use that system in terms of good breeding also. Thank you very much. I'm done, Chukka. Thank you, Abhishek.
Detailed presentation. So now we'll go for the lunch, and we'll come back around 1 o'clock. Then we'll have a detailed session on Aware data management platform. Jim will forward you the details at the time.
Thank you.
Yeah.
Do that on risk.
Whatever.
Risk.
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 In what?
What risk do you think we will face if we take these tools or the tablet or the page to enter data directly? Is there any risk or losing information
No.
from feed
No.
to the computer?
Actually, I have not got chance to bring that the tough pad what we have bought for our breeders. Anybody has a breeder has bought? Anybody?
You throw it like this in the field, you put it in the bucket of water, you break it like this, it will not break.
We have the tough pad. We have bought for all of ICRISAT breeders. I think I showed to few of my friends in the TL3 meeting. I can show you maybe in the afternoon. I'll show you. I'll bring it. It's only $1,000. No, but it has a long life. You see the study like this, I have taken as a biometrician because I will not go to field very frequently. And when I'll go, I'll keep it with care. Huh? But they are also sturdy. Like if you drop, they normally do not spoil, but they are very sturdy. And they have the matte finish on the screen, so you can see them in sun. I'll bring in the afternoon one, and the software has been installed in them also, so they are also very nice. In TL3, I have already requested PIN coordinator that with the partners, I am responsible for implementation of IBP tools and this tablet. I'll make it sure that all of us collect all data. If you see that the point we got the electronic data capture, we'll make it sure. No problem.
Then your life will be easier.
Yeah, when I get that kind of data, my analysis will be at least
Less
easier.
working.
Any other question? Is it okay? Yeah, it's for everybody. Any other question? I hope no.